inside every living thing, inside you, right now, there are trillions of microscopic machines hard at work. We call them proteins, and they do, well, pretty much everything. They carry oxygen, they fight off diseases, you name it. But here's the real secret. A protein's power isn't just about what it's made of. It's about the incredible, mind-bogglingly intricate shape it folds into. It's like microscopic origami, and its entire job depends on getting that final fold just right.
So that brings up a really critical question, doesn't it? If everything hangs on getting the shape right, what happens when that folding process goes wrong? How can one single tiny mistake in this molecular origami lead to a disease that can be absolutely devastating? Okay, let's dive in and find out.
Just look at the difference here. On one side, you've got a healthy red blood cell. See how it's smooth, disc-shaped? It's perfectly designed to just glide right through your veins. But on the other side, that's a cell that's been warped into a rigid, sticky crescent shape. That's a sickle cell. And those are the cells that clog everything up. This incredible transformation is the direct result of just one protein folding the wrong way. To really get a handle on this, we're going to look at probably the most famous case study there is, sickle cell anemia. It is the perfect, powerful example of how a massive biological catastrophe can start with the absolute smallest mistake imaginable. And this is the part that's it's astounding. Think about the scale of this error. The hemoglobin protein is this long chain made of hundreds of amino acids. But in sickle cell disease, just one of them gets swapped out. A single glutamic acid is replaced by a valine. That's it. And that one tiny change is enough to completely warp the protein's shape, which then causes millions of them to clump together and totally distort the red blood cell. It's this catastrophic chain reaction, all from one little type.
So if we want to understand how things can go so wrong, we really have to start with how they're supposed to go right. I mean, how does a protein pull off this magic trick of achieving its perfect shape? Well, it happens through this stunningly elegant four-level process.
Let's walk through this blueprint together. It all starts at the primary level, and then it builds in complexity, going through secondary, tertiary, and for some proteins, a final quaternary structure. Each step is built right on top of the one that came before it. Everything kicks off right here at the primary structure. And honestly, it's pretty simple. It's just the linear sequence of amino acids all linked together, like beads on a string. It might not look like much of a structure yet, but don't be fooled. This exact order contains all the information needed for every single fold that's about to happen. This is the fundamental code. Okay, moving on to the next level. Almost immediately, that long string of beads starts to twist and fold in on itself. Little local sections of the chain start forming these simple, really regular repeating patterns. This is what we call the secondary structure, and it's all driven by hydrogen bonds forming between atoms in the protein's backbone. And the two patterns you're going to see most often are the alpha helix, which is this right-handed coil, kind of like a spring, and the beta pleated sheet, which has more of a zigzagging folded paper kind of look. Most proteins actually have a mix of both of these structures. All right, now we get to the main event, the tertiary structure. This is where all those little helices and sheets we just talked about fold up on each other to create the final complex three-dimensional shape of a single protein chain. This compact globular form, this is what allows the protein to actually do its job. And finally, some proteins take it one last step further. The quaternary structure is what happens when multiple already folded protein chains, we call them subunits, come together to form one big functional complex. And this brings us right back to where we started. Hemoglobin, the protein in our red blood cells, is the perfect example. It's made of 4 separate subunits that all have to assemble perfectly into one working machine. Okay, so we know what happens. But the big question is, why does it happen? A protein doesn't just, decide to fold one day. It's actually being guided by these incredibly powerful fundamental forces of chemistry and physics. It's like an invisible hand that's constantly pushing it toward its final shape. And the number one driver of this whole process is something called the hydrophobic effect. See, our cells are mostly water, but some amino acids are hydrophobic, which literally means water-fearing. Just think of oil and water. They do not mix. Well, the hydrophobic effect is just this really powerful tendency for all those water-fearing parts to get away from the water. So what happens? As the protein starts to fold, all of those hydrophobic amino acids just spontaneously collapse into the center, forming this dense core. This move shields them from all the water that's around them, and in doing so, it locks the protein into its stable, compact, and functional shape. It's pretty much the most important force in the game. But, the cell doesn't leave a process this critical entirely up to chance. It actually employs a team of assistants called molecular chaperones. These are basically other proteins whose entire job is to help new proteins fold correctly. You can think of them as the quality control managers of the cell. These chaperones have a few really key roles. First, they'll bind to a newly forming protein to keep it from folding incorrectly or just folding too early. They also stop proteins from clumping together into these useless, unwanted aggregates. And this is pretty cool. They can even help unfold a protein that's misfolded, giving it a second chance to get it right. Which, of course, brings us back to the dark side of this whole story. Because even with all this complexity and all this quality control, things can still go wrong. And when they do, the consequences of a misfolded protein can be absolutely catastrophic, extending far beyond just sickle cell anemia. Alzheimer's, Parkinson's, Huntington's disease, even mad cow disease, all of these devastating neurodegenerative conditions are now thought to be caused by misfolded proteins that clump together into these toxic aggregates in the brain. For these diseases and so many others, getting the fold right is truly a matter of life and death. Now, for decades, scientists have had this holy grail to be able to predict a protein's final 3D shape just by looking at its primary sequence of amino acids. But this challenge has been unbelievably difficult. I mean, the scale of the problem is it's truly astronomical. This right here is the heart of the puzzle. It's called Levinthal's paradox. See, a protein has an astronomical number of possible shapes you could theoretically fold into. If it had to randomly try out every single one to find the right shape, it would take billions upon billions of years, literally longer than the current age of the universe. But we know that proteins fold in milliseconds. So what gives? Well, as Cyrus Leventhal himself figured out, it just can't be a random search. The process isn't random at all. It must be a guided pathway where the protein moves through very specific intermediate states to get to the finish line super efficiently. The way scientists think about this today is as a folding funnel. Picture this. The unfolded protein starts at the very wide top of the funnel. It has really high energy and lots of possible shapes it could be in. But as it folds, it's constantly guided downhill, energetically speaking, toward a narrower and narrower set of possibilities, until it finally settles at the very bottom in its one single, most stable functional shape. For 50 years, this protein folding puzzle remained one of the grand challenges in all of biology. Then, in 2020, something incredible happened. An AI company called DeepMind unveiled a program called AlphaFold. It's an artificial intelligence that can predict the 3D structure of proteins just from their amino acid sequence with absolutely astounding accuracy. It essentially solved the problem from a computational standpoint, a total game changer. This leap forward has changed absolutely everything. It really feels like we're on the verge of finally mastering this microscopic origami. And the amazing part is, the tool that's finally letting us do it isn't a more powerful microscope. It's an entirely new and powerful form of intelligence itself.